gene linkage and recombination, which is kind of mixing everything that we've been doing up to this point together with the dihybrid crosses and the gene mapping. And today will be the last day with all this. So I gave you, it's kind of a challenge problem. We'll see how it goes. If you are good with this stuff, you really don't need to watch the video. If you're not so good, you're teetering on the edge, you might want to watch it for a little bit and then, turn it off if you feel confident. So I would suggest that you use a separate piece of paper to answer the following questions. You can try a Google Doc if you want to. I would probably just get a piece of lined paper out, write my answers on that, and then take a picture and submit it if you have that option available to you. So you're doing a genetics experiment with the fruit fly in the pea generation. You cross two true breeding flies. The female parent is brown and wingless, and the male parent is black with normal wings. I should say male. When I did it, I must have made a typo there and didn't catch it last year. All the flies in the F1 generation are brown and have normal wings. Indicate the alleles associated with the dominant phenotypes by uppercase letters and alleles associated with recessive phenotypes by lowercase letters. Assume the genes are not found on a sex chromosome. Indicate the color alleles as capital B and lowercase b, and the wing alleles by the letters capital N and lowercase n. So the genotypes of the flies in the pea generation. So we have, they're homozygous because they're true breeding. So regardless of what we do with them, we know they're either going to be two capitals or two lowercases. One parent is going to be brown and wingless. The other parent is black with normal wings. And our children all have brown, normal wings. So that would mean that brown and normal wings would be the dominant traits here. So if I have a brown parent, then that would be the capital B. And we know they're true breeding, so that's why I have two capital B's, because they have to be homozygous. And they're wingless, which is going to be our recessive trait. The other parent is black with normal wings. So there's my parents. And it doesn't matter which one you have labeled as male and female, because this is not on the sex chromosome, which it tells us right here. It's an autosome, so it doesn't matter who you have as male or female. B, the genotype of the flies in the F1 generation. So my F1 generation is going to be all hybrids. They're crossed. So I'm going to have a capital B, a lowercase b, a capital B, and a lowercase n. That's all that I have to give for my parents. This parent's obviously going to get the capital B, this one will get the lowercase, and this one will get the uppercase, and that one will get the lowercase. You now take an F1 female and cross her with a true breeding black wingless male. This male's genotype is, so he is black. We know that black is recessive. And he is wingless, which is also a recessive trait. So even if they didn't tell us that he was true breeding, it wouldn't have mattered because his genotype is all recessive, so he has no option but to be homozygous and recessive. You count 1,600 offspring in the F2 generation. That's a lot of counting. If the wing and color traits were linked and no recombination occurred, you would expect to count number of brown-winged flies, black-winged flies, brown wingless flies, and black wingless flies. So you would probably do some type of a Punnett square. You could do a dihybrid cross there and do the 16 blocks. I'm going to do the shortcut version of it. And let's see, so my cross then, I'm going to be using that female. Cross with female up top. I'll put my male down the side. So brown winged flies. So if recombination does not occur, you would expect then either to be black winged flies or brown wingless flies. So you'd have 0, 800 here, 800 here, and 0 down there. Now, whenever you would actually go through and look at the F2 generation, that's not what you'll get. You'll get numbers that look like this, which is what our Punnett squares would be showing up as, because these traits are going to be connected. So what is the genetic distance between the color and wing genes? So we're looking for the recombination frequency. So you're going to take your recombinant traits, which is your 2 smaller traits, and you're going to divide them by all of the possible outcomes. So I know a brown winged fly is going to be a recombinant, and a black wingless fly is going to be a recombinant, because those are not what our parents were. So you're going to take 85 plus 75, and you're going to put that over top of 85 plus 728 plus 712 plus 75. So we'll divide that out, multiply your answer times 100. And you'll get about a 10% recombination frequency. Letter F, a series of fruit fly matings shows that the recombination frequency between the gene for wing size and the gene for antenna length is 5%. The genetic distance between is 5 centimorgans. List all possible recombination frequencies between the gene for color and the gene for antenna length. Use the recombination frequencies you found in Part E for the color and wing size. You will be creating a gene map for the 3 traits. Okay, so they tell us that the recombination for antenna length and wing size is 5%, and we know that the color and wing is going to be 10%. So we have, and you can make up your own letters for your traits. So I'm just going to put WS is going to be wing size, which is the same as, I believe, wing genes. A is going to be for the antenna length. And I'll use C for color. So my possible options that I have when creating this, I'll go ahead and draw just a line here. I know color and wing genes, so I'll put my wing size down here, and my color is going to go over here, and I'm going to say that this one is at 10%. Move it up a little bit. And then we know that antenna length is going to be at a 5. So I'll put the A right here, and I'll say, what is it, wing size? That's going to be 5. So I would have a 5% difference between both of them. That's one possible option. Now there is another way you could go about doing this because we're not sure really where the antenna length is between color. We don't know if it actually is just 5 or maybe it is more like 15. So we could create another map where I have color, wing size again. We know this is going to be 10, and we know that the antenna length is 5 away. So I could create this one down here, put an A over here, and make this 5. So then the length between antenna and color would be 15. Because we don't know exactly where antenna lies, we have to include both options in that because it could go either way. Does that make sense to everybody? So that's it for question number 1. Question number 2. Figure out a new way to erase this. You are working with a hypothetical fly and have found color and wing mutants. Preliminary work indicates that the mutant traits are recessive and the associated genes are not sex-linked. But beyond that, you have no information. You first look at 2 genes, each with 2 alleles, capital B or lowercase v for body color, capital W or lowercase w for wing surface. I made another typo. The red body phenotype is dominant to the yellow body phenotype, and smooth wings are dominant to crinkle wings. For A, you cross a true breeding yellow-bodied, smooth-winged female with a true breeding red-bodied, crinkle-winged male. What will the phenotypes of the F1 progeny be? So for A, I'm crossing yellow-bodied, smooth wings with red-bodied, crinkled wings. Red is dominant, smooth is dominant. Okay. So yellow body is going to be the recessive trait. So we know that one parent is going to be this, the other parent will be that. And you have to figure out your parents before you can figure out what their kids will be. Otherwise, you're going to get yourself all confused. And then the yellow body has smooth wings, which are a dominant trait, so it's going to be capital W. And the other guy has crinkled wings. So my F1 generation then is going to be capital B, lowercase b, capital W, lowercase w. So they're all hybrids. What will the ratio, or you cross several pairs of F1 siblings, what ratio of phenotypes do you expect in the offspring? Make a dihybrid Punnett square. If you're making a dihybrid Punnett square and you're crossing 2 hybrids, so I'd be crossing this genotype with the same thing, because that's what all of these flies are. Anytime you're crossing 2 heterozygotes in a dihybrid cross, as long as they are heterozygous for both traits, then that is going to be a 9 to 3 to 3 to 1 ratio, hypothetically speaking. Does it always work out perfectly like that? No, but when we're just talking sciencey stuff here, in our minds, that is kind of what we should have. And what we're saying is that we're going to have more that are dominant than we will have recessive. The actual data is shown below, so this is what really could happen. Does this match your expectations? If not, explain why. There's a difference in the expected and the actual numbers. So when we're looking at this, we're looking for our dominant traits, which would be red and smooth. So this number here would match up with our 9. And we know that yellow and crinkled is going to be recessive, so this would match up with our 1. And then these numbers in here would have to be our 3 to 3. So does it match up perfectly? No, not perfectly, but it is awfully close. It doesn't match up perfectly because these two numbers in here should hypothetically be the same, and they're not. But that's the only way it doesn't match up. Moving on to letter C here. To determine the recombination frequency of these two genes, you perform several crosses where you cross an F1 from part A with a yellow-bodied crinkle-winged fly. So you're doing a test cross now. You get the following results. What is the distance between the genes for body color and the wing surface in map units? So there we have the same table, but this is whenever they did a different cross. It's not crossing two hybrids this time. So to determine how to do our recombination results here, we are going to take our recombinant traits and we're going to divide them by our total number of crosses. So our recombinant traits are the ones that you have the least amount of. So that would be our ones in the middle here. These would be our hybrid versions because they are different from our parents. I'm going to move over here just so you can see it a little better. For letter C, we're going to take 102 plus 98. We're going to divide that by 396 plus 102 plus 98 plus 404. And you're going to multiply that by 100 so that it turns into a percentage. And that's going to give us about 20% chance of a recombination. So in math units, it would be 20 UCM. So you decide to turn your attention to a different gene, one that controls wing length. This gene has two alleles, capital L or lowercase l, where long wings are dominant to short wings. Remember that the red body phenotype is dominant to the yellow body phenotype. You again make two true breeding lines. So I'm just going to underline real quick my dominant traits. A red-bodied short-winged male and a yellow-bodied long-winged female. And you get all red-bodied, long-winged children in that generation, which makes sense because you crossed two homozygous parents. They're homozygous for both traits. So all of our babies are going to be hybrids where they have this genotype here. I can't remember what we used for the color. Body color was a B. To determine the recombination frequency between these two genes, you perform several test crosses where you cross an F1 from part C with a yellow-bodied short-winged fly. And we're always doing it with the recessive traits because we know that if there is a dominant allele in there, it's going to show. You cannot do a test cross with a heterozygote or with a homozygous dominant. It has to be homozygous recessive crossed with that unknown person. You get the following results. What is the distance between the genes for body color and wing length in math units? So we're going to go with the two that are not our parents. Normally they're going to be the ones that have the smaller numbers here. So in this case, it's that. So we're going to take for part D, we're going to do 45 plus 55 over 45 plus 460 plus 440 plus 55. Multiply that out times 100. And you should get about 10% or 10 cms. And that's going to be our math units. Oh boy. All right. Given your answers to part C and D, what can you say about the relationship between the gene for wing length and the gene for wing surface? So our gene for the first one, part C, was 20% or 20 map units away. For part D, it was only 10. So you would expect that the gene for wing length and the gene for wing surface to be linked, and the distance is expected to be either approximately 10 cms or 30 cms apart. Based on the preceding, what are the two possible arrangements for the three genes? Indicate the map distance between the genes for each arrangement. We're going to create a gene map. All right, so for wing length to wing surface. Let me erase this here real quick. Again. Okay, good. So our three genes that we have are wing length, we have surface, and what was our last one? Color. So we got to go through and make up our letters for each one of those. So wing length, we're going to use the L. For color, we use an R, and for the wing surface, we'll use an S. So this one was 10 for D, and that was talking about the distance between body color and wing length. Color and length is 10. And then we know that in part C, we were talking about the wing surface in relationship to body color. And we said that that one was 20. So body color, we're going to wing surface. Surface is S to color, which is R. So that could be one potential correct answer. Or we don't know if the wing length is actually that close to surface because right now we're saying that it's only 10 away. So our other option would be to put the R here, S over here, and say that this length is 20. I know it doesn't look like it's 20, but we could, we have our numbers labeled in there. Color to there, and then length would be down here at 10. And then that would put the surface to the length at 30, which was where I came up with those numbers just a second ago when I was talking about it. Okay, and now we are on to, I think, question number 3, which is the last question. I think, and thank goodness. It's a shorter one, I think. So while working with a type of beetle that is normally smooth, large, and white, we have 3 traits there, you discover 3 mutations that lead to the recessive phenotypes bumpy, small, and gray, respectively. You cross true breeding smooth white beetles to true breeding bumpy gray beetles and get all smooth white beetles. You then cross the F1 beetles to true breeding bumpy gray beetles. And after analyzing 800 F2s, calculate the map distance of 5% of 5 map units between the smoothness and the color loci. What are the 4 phenotypic classes you got in F2? And about how many of each did you get? Your total should be 800. For body types, use capital B and lowercase b to designate the alleles. For color, use white and gray, use G or lowercase g. And for size, use an L. So we didn't actually talk about size at all whenever we're talking about our F2s. So we really don't even need to worry about putting those L's in there because it only told us what the kids were, that they were bumpy and gray. So they're just giving you extra information here. You don't need to worry about whether they're small or whether they're large. We just know that they are bumpy gray beetles. So for a smooth white beetle, smooth is going to be what? Bumpy gray beetles. Across the smooth white beetle, the true breeding bumpy gray beetle. And I got all bumpy gray beetles. So first we need to figure out our genotypes. So you're going through and you're trying to figure out which ones are dominant and which ones are the recessive form there. So we know that gray is going to be the dominant trait there. So I'm going to give one capital G to both of those. We know white is going to be the recessive trait. So white is going to have 2 lowercase g's. And that means that the ones who are gray are going to have to be 1 of each. Since we're talking about that F2 generation. Now in order to be smooth, let's see, that's going to be a recessive trait. Right? Oh, whoa. Sorry, F1 generation are smooth and white. I need to back up a little bit here. So there was mom, and dad would have been lowercase b's, capital G's. So our F1s would have been heterozygous to be bumpy. And you cross the F1 true breeding beetles to the smooth or the bumpy gray beetles. That's 800 F2 to calculate the distance. Examines between the smoothness and the loci. Hold on 1 second here. All right, so we got capital B for smooth, lowercase b for bumpy. It's going to be all lowercase ones. Smooth is going to be heterozygous. So how many of each phenotype? So we got to look and see what our parents were there, and we know that we're going to have less for our recombination if they are not one of the parent traits. So roughly speaking, you want your numbers for the bumpy and the white ones, and for the smooth and the gray ones to be significantly smaller. We're trying to add up to about 800 here. So our numbers for our parents should be about 380. And our numbers for our children should be about 20, showing that they are recombination children. That last one there, you cross 2 true breeding parents to get all F1 beetles that are large and smooth. You cross the F1 true breeding small bumpy beetles to get 28 large smooth beetles, 19 small smooth, 16 large bumpy, 237 small and bumpy. What are the genotypes and phenotypes of the 2 parental beetles in the P generation? So we know that they're going to be homozygous for both traits because it tells us that they are true breeding. Across F1 true breeding, small bumpy beetle. So we would write small bumpy for one of our phenotypes here. And we need large smooth beetles to be our other one. If they are large size should be dominant, so it's going to be capital L's. That's going to be lowercase l's. Bumpy is lowercase. It's going to be lowercase e's, capital E's. And that would be it for this activity here. All right, thanks for hanging in there with me. I know that was a bit of a long one.